Okay, hi everyone. Thank you so much for joining me today. It is 2 p.m. This is a little bit earlier than usual. We're trying some new times. But I just want to say thank you for joining us. My name is Danielle Snowflack, Dr. Danielle Snowflack. If you joined me before, welcome back. If not, I am the Senior Director of Education at Edvotec, and today I am so happy to be able to share with you this biotechnology training workshop in the comfort of your own home. This one is going to be fun. It's going to be very different in that we're going to be really trying a bunch of things and exploring, and some of these things I haven't even tried yet. So come with me for the journey. We're going to have a great time. For those of you who are unfamiliar with Edvotec or you're new to our live streams, Edvotec is the biotechnology education company. We were founded over 30 years ago by Dr. Jack Trichian, who was a professor of biochemistry at Georgetown University. And our company was born to translate exciting innovations in the world of biotechnology for the classroom. And this is important because if we think about the period in time when this company was founded, it was a boon in biotechnology. We had molecular cloning techniques, which were becoming very common, the cloning of insulin, the development of the polymerase chain reaction. And these were all really great things, interesting things, and exciting techniques. And how could we bring those into the classroom to excite students? So hi, thank you so much for joining me, Karen. It's always nice to have someone in the chat to say hi. But we're here now to work with our educators to help bring in hands-on learning experiences, to demystify science, and to really foster the next generation of science learners. And so we really want to make biotechnology accessible to everyone, to the lab, to research spaces, outreach spaces, biohackers, and our educators. We are recording the demonstration, and the slides will be available on our website. If you want to be notified when they're posted, please fill out the form. Let me paste that in. I am pasting the link to the form. That form will share your email address with me. I will be able to send you the slides. And if you would like a professional development certificate for participating in today's workshop. The link is only going to be alive for an hour after the presentation. So please be sure to fill it out before 4 o'clock Eastern time. So today, hold on a second. My window capture is not working right. There we go. Okay, that's the slide we're supposed to be on. Our next slide is talking about the scientific method. So today we are talking about the scientific method. And I think there's a little debate on whether or not teaching the scientific method is right in classrooms, because it's not necessarily the process through which people are doing science all of the time. Like every experiment we do doesn't follow the same set of steps. But I think it's a really great logical framework to get us to start thinking about the best way to design experiments and to think about the research we're doing and to answer the questions that we are asking in the laboratory. And so this is a logical process that we can use to start framing the way we're thinking about science, we're thinking about our experiments, and we're moving forward and doing them. And so you're probably teaching this right now. We are in the beginning of the school year in many places, or maybe you just taught it. So this could be information for next semester or next year. But I do think it's important to learn about the scientific method and to really think about it. And so the word itself, science itself, is derived from the Latin word to know. And it makes sense, right? Because the scientist's quest is to know and to learn more about the world around us. And so we have this framework, the scientific method framework, where we observe the world around us to start thinking about questions that we can ask. We do research to learn about existing knowledge and how that can build and help us answer questions. With this information, we start to make a hypothesis or an educated guess that we are going to test through rigorous scientific experimentation. Once we have prepared and we have our hypothesis, we then design the right experiment to answer our question and to generate our data, the pieces of information that we're generating are our data. And then we report out this interpretation. And this picture that I have over here here. I can never get my hands to go in the right direction. But this framework that's on the slide, this is like a figure that you'll often see in your textbooks. And it has the scientific method as a linear process. You start with your observations, you enter through your reports, and that's it. But that's not really how I think about the scientific process and the scientific method. I personally think about it more like this, like a circular process, because science is constantly building upon the science that came before it. And so this builds from previous knowledge and observations, previous scientific reports, and then we take that information and we contribute that information to the body of scientific knowledge, and then that moves forward and we start again and we ask more questions. And even negative results contribute to that. And I think that's important to note, because I think that sometimes when our experiments don't work, I think that, that can be disheartening to students. But failure and negative results are part of the scientific process. They add to that knowledge of what we know and what we don't know. And then again, we can use that to ask the right question the next time. And I think that we do want to make sure that we do not necessarily consider negative results as failures. They are just more data that allows us to ask better questions the next time. And it's very valuable. So today, like I said a little bit in the introduction, we are going to play. We are going to use a bunch of stuff I found around the house to explore fluorescence and to explore the scientific method. And so on this slide, we're going to be answering a question that a teacher asked me a few months ago. What else can I do with the True Blue transilluminator? It's a large part of my equipment budget, and I was wondering if I could do more than just look at gels. And so you have the True Blue on the slide. It's flashing on and off. Basically, that is the transilluminator. We use it to look at our results on our gels. But is it a unitasker? Is it a piece of equipment that you can only do one thing with? And I say 100% no way. This is a piece of equipment that is super useful in the lab, and we are going to ask a bunch of questions and test them using the scientific method. And so if you get the slides, if you ask for the slides by clicking on that link, you will get this presentation. And that purple underlined part is a link to a blog post where I performed some of these experiments previously. So if you don't have a True Blue 2, you can likely do these experiments with a handheld UV light box, or a handheld UV light, or another UV box. I haven't tested this though, so I would recommend testing it first before you try it with your students. But in this case, we are using the True Blue 2. The True Blue 2 is a perfect tool for the classroom because it has both a blue and a white light feature. So already you're able to do more than just like a cyber safe. The white light is perfect for visualizing electrophoresis or autorads or anything else where you need a bright white light. But a lot of its power comes from the blue light function, which allows us to observe fluorescence. All right, so here is a slide. I'm going to go over to my webcam. Let me turn this extra light off. We're going to be going back and forth between light and not light a lot of times because the fluorescence looks better when my room is dark. And so, as I mentioned, you might be using the True Blue already for its intended use, which is a blue light transilluminator for imaging DNA separated using CyberSafe containing gels. CyberSafe is a classroom-safe fluorescent molecule that's going to specifically bind and label DNA. And so you can see that in the picture here with the gel, and you can see that, even if we just add on the slide you're looking at, you can see that even if we just add this is the wrong slide capture again. Sometimes my software forgets where I am in the slides, but here's where we're supposed to be. And so CyberSafe is normally used to visualize DNA. It binds to the double-stranded DNA, and only when it's bound to double-stranded DNA does it glow bright green. And I actually wondered whether that was true. So whether CyberSafe only glowed in the presence of DNA or whether it would glow just in the presence of water. So I tried that experiment. And what you can see is that in the slide where I have the three little tubes, If I have cybersafe and buffer, or buffer and DNA, I don't get any fluorescence. Only when I have the DNA and the buffer and the cybersafe do I get green. So there's already a little glimmer of the scientific method, asking questions, making observations, and getting results, right here on the first slide. Our True Blue 2 is perfect for doing our GFP experiments. So if you're visualizing the fluorescence of GFP, the green fluorescent protein, it's excited by blue light. In fact, in the jellyfish, GFP is excited by blue light, which is generated, and we first identified that protein in a jellyfish. It's excited by blue light. We can see here glowing. We have some untransformed E. coli and some transformed E. coli. They're glowing bright green on the True Blue 2. We have just some extracted GFP. That's from our bioprocessing experiment, which you may be doing. And so you can see that's glowing bright. It's beautiful color green. But, let's start exploring other things than what we normally look at with our transilluminator. Let's start exploring other ways to, other fluorescent molecules. And so we're going to start doing some chromatography. And so you may have joined me and we're going to do this chromatography from markers, pigments. And so let me put this here for now. And maybe like my crazy, you're not even going to see my crazy selection of markers, but we have our highlighters, we have our markers here, and we're going to use paper chromatography to analyze markers and determine which of these markers have fluorescent dyes present. And so you may have joined me for my previous workshop, which was Sweet Science, where we talked about analyzing food dyes by separating them by color. One of the techniques we used, so let me go back to my face cam so you can see me. So one of the techniques we used was paper chromatography. We're going to use that technique today to do some of these experiments in our first exploration of fluorescence. And so the structure of food dyes are shown here on the slide. And at first glance, they're similar molecules. They're all around the same size. They all have these aromatic rings. But when we look more carefully, their chemistry is actually different. We have some different bonds, some double bonds. We have different functional groups on the sides of these molecules. And all of these are going to affect the physical properties of the molecule, their color, the way they separate in solution. And this gives them, unique identities and unique reactions when we look at them in solution. And so, as we showed in our previous experiment, we can separate dyes like these using paper chromatography. So these are food dyes, but dyes are also used to infuse our fabrics with colors. And so if you look at your garments, they can be nearly any color you can imagine. And actually, let's go back to our webcam here. And here are just a selection of colors that are created by dyes in markers. And so, our garments are dyed colors by taking mixtures of dyes and chemically binding them to fabric. And even food coloring, like we looked at the last slide, these can be used to dye fibers that are built from proteins like wool or silk. And so the ways these dyes work is that the molecules absorb most of the colors in the light that are bouncing on it, that are coming in contact with it, and most of this light is in the visible light spectrum. So if you're wearing a yellow shirt, like the one on this slide, the dyes that are bound to the fabric are absorbing most of the light, except for that yellow light. That yellow light is going to be reflected off of the dyes in the fabric, and it's going to reflect to your eyes where you can perceive the color. And that's how dyes are going to create color on our fabric. But if we look at a safety fabric, it's really hard to photograph. These are the best picture that I could find, but it's really hard to photograph because safety fabrics like this one, they're infused with a fluorescent dye that reflects UV light back to our eyes. And that can make it appear even brighter, which allows workers and crossing guards or cyclists to stand out. And so what I'm interested in exploring in this workshop today, in this first exercise, is fluorescent dyes. So where do we find them? How do they work? And can I analyze them in solution? And so the first thing to think about is, what is fluorescence? So this is the research. We're starting to make observations. And if we're thinking about this again in the framework of the scientific method, we have some information. We've done an observation, these fabrics that seem bright. Let's do a little research. So what is fluorescence? So fluorescence is the phenomenon through which some substances absorb light at one wavelength and then emit light at a second wavelength. And the most dramatic results are going to come when we can excite a substance using energy, like UV light or visible light, and then actually see that fluorescence with the naked eye. And so in this picture, we can see some fluorescent minerals, which are compounds within a rock that can fluoresce when excited with UV light. And while these minerals can generally be found anywhere in the world, one of the most famous places is located at the Sterling Hill and the Franklin Mines in New Jersey. And I went to these places a lot as a kid because they were right around where I grew up. And these fluorescent minerals are just so much fun, and it's so cool to pick up a rock and to just shine a light on it and watch them shine. So naturally occurring proteins can be fluorescent as well. And, our favorite is GFP. Again, let me bring the GFP back into frame while we're here. We may as well look at the GFP while we're talking about GFP. And so naturally occurring proteins can be fluorescent as well. This GFP is our fluorescent protein that fluoresces green when it's excited by blue light. But the thing is, using these amazing biotechnology techniques, we could scientists and researchers sequence the GFP gene, which allows us to clone it into other sources, but also knowing that sequence, we can change the sequence to create a full rainbow of colors. And these can be used in a myriad of ways in the science lab. And I just love this picture because it shows you what bioengineering can truly do when we get the sequence and we understand what the structure of a protein is doing. And we can really, visualize what is going on and analyze it and go forward. And so you can see we have some proteins here that are derived from GFP. RFP is another fluorescent protein. And that one was able to be modified to create even more colors. And we can see their excitation wavelength, which is the specific wavelength of light which allows them to fluoresce. And so one of my favorite techniques in the lab from when I was a grad student was fluorescent immunohistochemistry, where we can actually label proteins within individual cells with fluorescent antibodies. And when we shine a laser on them using a microscope, we can actually see where these specific proteins are located within the cell. And so that is a super amazing technique that has allowed researchers to move forward in understanding, what is happening even at the smallest spots within our cells. Okay, so now that we've done a little research on fluorescence, let's go back and make a few more observations about, dyes and about fluorescent markers and, take these observations and design an experiment. Okay, so I was talking about markers. Markers are the dyes that I am using. We talked about fluorescent dyes in clothes and how fluorescent works. Now, when taking notes, I often use just a plain old highlighter. I'm not naming brands, but you can see them here. But, we've all got a bunch of highlighters around. And when we use these markers to highlight passages, they are bright, they are neon colored, and they really stand out. And so I want to take a bunch of these markers to look more closely to determine specifically what makes them so bright. And so I swatched out a bunch of markers here on this page. I have highlighters, I have permanent markers, and I have watercolor markers. And what we can see is some of the inks definitely pop off the page. Again, it's harder to see on the paper, I think, because of the camera. But like when I look at them by eye, the yellow really pops off the page. We have this bright pink. We have this bright orange. Those colors are popping. And I wonder whether these eye-popping colors are suggesting that the samples have fluorescent inks present within them. And how can I determine that experimentally? So this is the fun part. So if anyone sees a color here that they'd like me to specifically analyze, I did not have a yellow permanent marker to test, but if anybody sees a color here that you want me to test, let me know, because I will swatch out that color specifically. I have two vessels that I can use to perform the experiment, and so we can swatch out two different colors and watch them separate and analyze them using this technique. So at this point, I think we've made enough observations to move to the fun part, which is creating and testing our hypothesis. And so there are a ton of questions that we can ask from our research and our observations. Is there something added to the fluorescent inks that make them extra bright? Do all yellow markers contain fluorescent dyes? Do all highlighters contain fluorescent dyes? Do other markers contain fluorescent dyes? And so we're going to analyze these markers using fluorescent paper chromatography. And so paper chromatography is going to use a paper matrix and a solvent to separate molecules based on their physical properties. At the end, we're going to separate these dyes into discrete zones or bands based on the physical properties of the molecule. And we'll be able to use our eyes to determine what combinations of visible light pigments are in here. And then we're going to use the true blue too to determine whether fluorescent pigments are present. And so if there's anyone who wants to see a specific color, let me know now and I will swatch that one out for you. Otherwise, I'll do the yellow because the yellow is super bright. I also have a fluorescein control that would go through the yellow. And then I'm going to do, I think the pink, because I have 3 pinks. That could be fun to do, unless there's another color that someone wants to see. And then I would do that color. So let's prepare for our paper chromatography experiment. So the first thing is that we're going to use a pencil, and we're going to use this pencil to draw a baseline. So this is just filter paper that I had. I've cut it into a rectangle that is going to fit easily into our vessel. Again, depending on the vessels you have at home, your size and your shape is going to be a little bit different. That's okay. This can be done with anything that you have available. I'm going to take my pencil and I'm going to mark the baseline, which is where I'm going to spot my ink. And this is where we're starting. And it's important to use a pencil when you're doing these experiments, because if we use the pen would also be able to be separated and to move through the paper using chromatography. So I'm going to start with the yellows, since no one has asked for a specific color. I'm going to spot my control dye onto this card. This is a dilute solution of fluorescein. I'll throw it on the transilluminator with this card when we're done doing the separation. But basically this is a fluorescent dye. You can't even see it because it's very dilute. I'm going to actually put a little F right here and mark where I'm starting all of my samples. And so I'm just going to spot a little on here, let it dry, spot a little more, because I want to have a really concentrated section of this dye right at the line. I'm going to do my yellow highlighter, so I'm going to do highlighter here, H. And let me just dab. For this, we're going to just dab, 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 dab until we have a really nice concentrated amount of ink there. I can even see it come through the back of the card. So we know it's really saturated at that spot. And then I have this watercolor marker. This is just a watercolor brush pen that I happen to have around. I'm going to spot, spot, spot. This one's a little harder to get a lot of ink on because it's got this brush tip, but we're just going to spot, spot, spot. And the thing is, we need very little sample. This is going to be able to separate really well. I'm using this technique. And so I'm going to then use the pink on the second card because I like pink and I have 3 of them. I'm not going to put the control on here. I think it would get too crowded. You do generally want to have your control on the same paper, but my vessel isn't, I don't think, large enough to get 4 across. So I'm going to do my highlighter here. I'm going to do my Sharpie here, and I'm going to do my watercolor marker here. And so here's the pink highlighter. Again, dab, dab, dab until I get a nice spot there. With the Sharpie, again, dab, dab, dab. And then with the watercolor marker, I want to make sure to get a lot of that ink on the page. And, we should even be able to see the spots through the paper to really ensure that they're saturated. So I'm going to let this dry now. And then I'm going to prepare I'm going to let these cards dry now. And then I'm going to prepare the solvent that we're using. So here, let me go to the next. We're going to let that dry. So we're going to make our solvent quickly. I didn't prepare it beforehand. I just have a bottle of water. I labeled it experiment water so I don't drink it. And then I have isopropyl alcohol. These are just what I happen to have around. And we'll talk a little bit more about solvents at the end if you want to explore further and ask more questions about what you're doing. I'm going to just use a conical tube that I have pre-marked as a device for measuring. I'm going to add 2/3 parts isopropyl alcohol, 1 part water. So we're going to have about a 66% isopropyl alcohol solution. You can change these proportions however you wish. But this is the experiment this is the concentration that I'm choosing. And I'll be honest in doing it because it's an easy concentration for me to measure easily on screen. But if you're doing this in the classroom, you're going to have access to a lot more sophisticated measuring devices than I do. And you will be able to, make a more controlled solution. So, let me measure the water out. This is going to be 5. So this will be our 1/3 part water. 1 part water to our 2 parts alcohol. And you can see how quickly I set this up. And that I think is going to be a big part of why you're going to love to do this experiment as part of teaching the scientific method, because the prep is super simple, super easy, super fast. You could run this experiment several times over the course of your class. So our samples should be dry by now. We've got them on here. You can either stand or tape your chromatography paper in the vessel so that the bottom is touching. And I'll move the camera so we can watch them separate. You want the paper to get in there. We're going to prop it up here. And then let me bring my camera down a little more. Let's see if I can, focus in on this pink one here. And so we're going to keep it focused there while the dyes are separating. And then I will talk a little bit more about what is going on inside of our paper. After a few minutes, we're going to analyze these results, because as you can see, this process is fast. We're already moving those dyes through the paper. And so in paper chromatography, we separate molecules using the stationary phase, which is our paper, and a mobile phase, which is our alcohol. The stationary phase is used to physically separate the molecules into different pools once we begin to develop our results when we add the mobile phase. And what does this mean? This basically means that the molecules in the sample are carried through the paper by the buffer. And so the molecules move through the matrix where they are gradually separated. The separated zones of the molecules are then visualized to let us identify the chemical compounds in the mixture. And we're talking about separating dyes here. Dyes are chemical compounds. Water is a chemical compound. Alcohol is a chemical compound. And so we're basically just analyzing these chemicals that we have present in this sample. And so as the solvent travels up the paper, it's going to bring the dyes up with it. But this is dependent on the solubility of the molecules in the mobile phase. More soluble dyes are going to travel up the paper faster with the solvent. And so we can already see that happening. Our dye front, is right around here. If you can see my finger, let me grab a pencil. So you can see the solvent front is right around there, and it's continuing to rise. And we can see that some of the dyes are moving really fast through the paper, and others are still down here by our baseline where we spotted and where we got started. And so insoluble dyes stay at the bottom of the paper. They're not dissolved by that mobile phase, and they cannot be moved through the paper. So they're going to stay at the bottom of the paper or move slowly. And the solubility of these molecules depends on the solvent. The solvent is generally a mixture of buffers and chemicals that are used in water that can move the sample mixture through the stationary phase. The solvents can be polar or nonpolar, and the actual nature of the compounds in our solvent are going to influence our experiment and influence the way things move through the gel. The stationary phase itself, so this paper, is also going to influence the separation of the molecules. In this case, we are using a cellulose-based paper, And the cellulose is polar. So that in itself is going to affect the way our dyes move through the paper. The matrix can capture some molecules that pass through the paper based on its chemical properties. But if we used a different matrix, like a thin layer chromatography plate that was maybe covered with something else, we could see differences there as well. Okay, so let's define a few key terms. We can see them here on the picture on this slide. Did my slide advance for you guys? No, it did not. Am I on the right? Oh, no, it is. Okay, there we go. So we're going to define some key terms. Then I'm going to take out our paper and we're going to mark them up. So if we look at our picture, we can see that once our paper strip has developed with the solvent, with our mobile phase, we're going to have spots on the gel. And these spots are the partially or completely separated zones of molecules that we see on our plate. Each molecule travels a distance from the origin, which is the location where the samples are spotted. So that's our baseline that we marked in pencil. The solvent front is the distance that the mobile phase travels through the matrix. So that's the maximum distance that anything can travel, because that's as far as the solvent has traveled. And so when I take these out, we're going to generally mark the solvent front and each of our spots with a pencil. And we definitely want to use a pencil again. You do not want to use a pen, again, because the solvent is still going to be wet and it can still move those dye molecules in a pen through the paper. And so if you want to bring some STEM in, if you want to do some math with your students, you can calculate the retention factor for each of these molecules. And the retention factor is the ratio of the distance the solute traveled to the distance that the solvent traveled. And so when the experiment is finished, you can make these measurements and you can calculate the different values for each of these dyes as part of your analysis. All right, so let me reorient the camera again to get an overhead shot. And so I'm going to pull out our samples, and we were running these for maybe 5 or 10 minutes, not 10 minutes, maybe 5 minutes. And you can already see how far it traveled. So here's our solvent front I'm going to mark right on both of these. I don't see anything visible here in my fluorescein control, but what I see here is a large bright yellow spot in both of these. Okay, here I kind of see this spot here. In the pink, I see a dark pink here. I see kind of a blob here. And I see this insoluble pink here. And then for our watercolor marker, I kind of see a long spot here. And generally, when I am calculating the retention factor, I kind of use the middle of the spot as the place where I make the measurement. All right. And so what we can see is that we do see clear differences in just the visible dyes that are associated with each of these markers. But we want to know whether our fluorescent markers have added fluorescent dyes in order to make them glow, right? So that's our question. And whether these spots can be separated out, whether specific spots fluoresce, because, you know, this experiment is all about using what we have to analyze fluorescence. All right, so let me turn this light out. I guess we'll look at one at a time. Just to, all right. And so what I have a smear of something on here, something fluorescent from my finger. Oh, you know what? It is something fluorescent from the paper. And so what we can see are some surprises. So in our permanent marker, we can see bright fluorescence in all 3 lanes. So in order to make that pink color pop, there must be a dye molecule involved that does put out light that is brightly fluorescent. Now, when we look at our fluorescein, what we can see is that most of the fluorescein is concentrated up here at the top. And again, that was a very low dilution, so we probably could have used more there. In our highlighter, look at that blazing spot. We can see so bright. And I think, and it definitely shows that part of the reason that the yellow highlighter is so much brighter than the yellow watercolor marker is because of the addition of a fluorescent dye. And so this is just a really simple and easy way to start exploring fluorescence. Let me give you the overview of all of those colors that I shared with you earlier. So I have this whole card that I spotted out. And let me move the camera up a little so we can get it all in one. And pretty much what you can see here is, in accordance with what we expected. With our yellow, we see a lot of really bright fluorescence with the marker, not really much with the watercolor marker. This is the highlighter. With our pinks, we can see blazing fluorescence in our highlighter and in our watercolor marker. The surprise to me is actually the Sharpie. I didn't expect to see any bright color there, and here I've got it. And then we can see the orange highlighter generally would have something fluorescent involved as well. But our blues and our greens don't. The blues and the greens have a bright color just by the nature of the pigments that were used. And so what we could do, if we go, let's go back to one of our chromatographies. Let's go back to the pink, because I think the pink had a lot of interesting colors there. Not only in the visible light, but in the fluorescent. What we do is we could take this data here from this experiment, and since we'd be in the context of our classroom, what we could do is ask another question. So, all of these are glowing slightly differently. On my camera, when I look at the camera, they all look pretty bright, but when I look at them by eye, this Sharpie has more orange in it than the other two. So how could we analyze which different pigments are in here? Can we separate them out into different pools? What could we do to really start to understand more about the nature of this mixture? And a great inquiry-based addition would be then to have your students take these samples and either change the proportion of isopropyl alcohol or change the solvent. So go from alcohol perhaps to salt, or a different mobile phase, and to see whether we get different results. And so do we get different separations? Do these bright fluorescent molecules look different when we change the solvent? Do we have a different paper, a different matrix that we could use? Does that affect your results? And are there different separations or retention factors when you use different dyes, different solvents? So that is the end of the first experiment that we're going to do. The second one we're going to talk about is chlorophyll. Let me turn some more lights on so I can see again. And so chlorophyll, our next demo is going to focus on plant pigments. So let me pull some of this stuff out that I'll be demoing. And so we know that chlorophyll A and B, which are the plant pigments responsible for absorbing light energy in photosynthesis, absorb light in the blue range. Furthermore, there are other compounds within plant cells, like beta-carotene or xanthophylls, that are pigments that are known to absorb light and emit in the visible, and we can see them in the visible spectrum. So what would happen if we extracted these molecules from plant tissues and visualized them using their True Blue 2? So my hypothesis is that we would be able to see the plant pigments fluoresce. And a lot of the researcher students here would do the same. If you're researching about fluorescence, you can do research about, different plant pigments and their excitation and emission ranges. Furthermore, if you're doing plant science, you can have your students expand on photosynthesis, about the process of photosynthesis and why everything absorbs in these specific areas and how that goes into the plant's process for building food. In my case, I went to my kitchen, I just grabbed some vegetables and I went outside to get some plants. I have this rose from a rose bush. I'm going to try the petals. I have these Brussels sprout leaves from dinner last night. I have a tomato and I have some spinach extract. This is actually from an experiment that we have at Edvotec. So this is actually going to be purified. What are we going to see? Are we going to get fluorescence? Who knows? Let's give it a try. And so what I'm going to do is I'm going to place small amount. First, I'm going to label my tubes. So let me start, with the rose. So I'm going to label that with R. And what I'm going to do is I'm going to place a small amount of this rose petal. Maybe I'll take a couple, I'll rip them into small pieces and kind of stick them in this tube. Maybe I'll take 2. That'll probably fit well in here. I've never tried this rose petal before, so we'll see how it works. I know green leafy things work, but I don't know about pink leafy things. But again, there are plant pigments present, so we can assume perhaps that it will fluoresce. I am going to add a solvent to this tube to help me extract this plant pigment. I'm going to use just plain old acetone nail polish remover. What is this? Does it say? It's mostly acetone. You can see there's a bunch of other stuff in it. We'll do a control to make sure that this doesn't fluoresce. I did test that. I do know this doesn't fluoresce, but you do want to have your students do the control to make sure that it doesn't fluoresce. And so I'm going to use this as a solvent. I'm going to add it to each tube. Now, if you don't have acetone, or you're not allowed to use acetone, you can probably use isopropyl alcohol, probably even ethanol too. I haven't tested that. I haven't tested either of those. I think the isopropyl should definitely work. I don't know about the ethanol, but I think that would probably work as well. And so I've got my plant tissue in here, and I'm just going to take a chopstick, which I happen to have, and I am going to use that to mash this plant matter. Okay. And it's hard for me to show you exactly what I'm doing without dumping this everywhere. So let me get a couple good mashes in here, and then I'll show you what the tube looks like. Mesh, mesh, mesh. Then you can see I've got mashed plant mixture. And then let me kind of push more of it down to the bottom. And you can see I have a dark pink color to that solvent. So that change in color lets me know that I have mashed and broken open these plant cells, and that these plant cells are, open, and I have extracted some of the pigments from them. And I am going to dump this out into a separate tube, so we don't have to worry about any of that plant matter. Look at that. It's a beautiful color, rose pink. I guess that's what we would expect. Let me put the R here. Next, I'm going to try these Brussels sprout leaves. They're dark leafy green. I have the spinach, which is also a dark leafy green. Will they be redundant? Will they look the same? We'll find out. So I'm going to rip off the hardest part because I don't think that'll mash particularly well. Let me stick these in. All right, let me get one more leaf. And again, this is a place to really encourage your students to experiment and to try things. And they might have something different at home they want to try. Spices, my understanding is turmeric, which I did not have at home. Turmeric is going to fluoresce, which is something I've never tested. It would be really fun to try in the future. And I'm sure there's a lot of other things around that your students could bring from home and see the fluorescence of. And again, I've got my chopstick. I'm trying to kind of like bring up that plant material and then mash it down. You really want to break open as many of the cells as you can. So I've actually never tried these Brussels sprout leaves. My expectation is that they'll work as well as spinach, which I have tried before. Which you do have in this purified pigment form. All right. So you can see I've mashed it. It is green. I try mashing it a little more. It mashes a lot easier with the tender leaves, if I'm being honest, than with these harder Brussels sprout cabbagey leaves. But let me transfer this to another tube again. I'm going to mark it with a B for Brussels sprouts and me. Here we go. Okay. And do you guys want to see the tomato? Let me know if you want to see the tomato while I am dumping this out. You know what? Let me mush this down a little more. All right. All right, and what we can see is we've got kind of a green with beautiful, okay. No one mentioned tomato. We've got time though, we got 15 more minutes. Yeah, I'll do the tomato. We'll see how it turns out. Again, I'm gonna take it, put it in a tube. Oh yeah, there's definitely enough time for it. We still got about 15 minutes, so let's just do it, right? We're just doing science. I have done tomato before. If you have seen my True Blue blog post, you will have seen that I have extracted from the tomato before. So this is one where I kind of do know, but the Brussels sprout and the rose petals are going to be new for me, which is exciting. It's always exciting trying something new, a little nerve-wracking, try it on camera because, you know, who knows how it's going to turn out. Oh, this tomato is definitely mushy. You know what? I'm just going to break it open. I'm going to add a little more acetone, and I'm not even going to try and take off the pulp. I used a larger tomato the last time, so maybe the cherry tomato is not as easy to do. Maybe it's more pulpy. But again, we're doing science, right? We're getting messy, you know, we're trying things. And, you know, I think that this is another reason why this kind of exploration is so much fun, because like we really don't have any preconceived notions, and we don't need to get a specific result. You know, this isn't an AP experiment. This is just something for us to do that is a fun way to think about science and a fun way to think about fluorescence, and to ask questions that we haven't asked before. All right, so let us bring this over here. Again, I'm going to get my True Blue back into the screen. The best thing is that you should keep it as dark as possible when you're doing it, you know, because it definitely makes it easier to see the fluorescence. And so I'm going to put my spinach extract on here. Let me peel this label off. It does not want to come off. All right, so I'm going to put my spinach extract. Next to that, I'm going to put my tomatoes, and then I'm going to put my roses, and then I'm going to put my Brussels sprouts, and they should all be right there. Let's get them in the middle of the screen. And turn on the light. Let's see what we got. Okay, this is so cool. So we know the chlorophyll, it's going to, with blue light, it is going to be excited and turn red. So that is the color of that chlorophyll turns. And so what we can see here, I'm going to point with my chopstick. Here, look at the lid. The lid of this vial has something fluorescent in it that is reacting with the light. So that's fun. And we'll talk a little bit about that in the next one. But look, we got this beautiful bright red fluorescence from our chlorophyll. The next was our tomato. So the tomato is also kind of red as well. Oh, wait, I forgot my control. Need a control. We always want to do good science. Let's get some just plain acetone in here. So when I did the tomato before, again, I used a different tomato. I felt like it was more of a yellow color. You know, let me see if I can get some of the extract out of the tomato, because maybe the other gushy stuff, that's the scientific word for it, gushy stuff, you know, is kind of interfering with what we can see. So I'm just going to use a pipette and bring that out into another tube. Leave the gushy. So here, this one is our rose petals. You can see we're kind of getting a yellow fluorescence. We have our Brussels sprouts. I think that they would be red if I had more pigment in there. It would be more red like that. It looks kind of orangey here. And then here is our control. Here is our tomato gush. So you can see that some of it is fluorescing, but really, okay, so now that I got the tomato extract away from the tomato pulp, we can see again, it's that kind of yellow color. And my guess is that the red pigment that is in the tomato is similar to the pink or red pigment that is in the leaf of the roses, and that's why we're seeing a similar color from those two. And so this is, again, a place to explore. And if you looked at the blog post that we have online, like I said, I have done some of this before, but I was curious as to what some of these pigments were and whether we could separate them using chromatography. And so I thought I would be able to see them, and I tried the experiment, and this is what I saw. So you can see here's the comparison between lettuce and tomato. The spinachy lettuce, green, dark leafy green, you can see again, is that really dark, bright red fluorescence. I did not have any spinach around to do this time. So again, the Brussels sprout leaves are that kind of green, leafy green. But you can see that, yes, the pigment does move through the paper. At that time, I did it with isopropyl alcohol. But again, if we took these pigments, spotted them onto paper, and did the chromatography, what would we see? And how would this separate? And so I feel like if we did a little more research and development, we could get superior results and really separate some of those pigments out, calculate our retention factors, and then go from there. But sometimes, like what we're doing today, it's just fun to squish stuff and to see what turns out. And so while I was in my kitchen, I realized that, I have a lot of plant-based oils. So from baking, from cooking, I had several plant-based cooking oils. I had soybean oil, corn oil, and olive oil. And so I wondered, these are plant-based oils. Are these also going to fluoresce? And if they do, that means that some of the chlorophyll or other plant pigment remained in the oil after the plant samples had been macerated and the oils extracted. And so here is water, which I'm going to consider my control for this. Our water does not fluoresce, which means the water doesn't fluoresce, the tube doesn't fluoresce. And now when we think of olive oil, we often often think of it as like a darker green oil, and so it makes sense that there would be some fluorescence there. And what we see is that is true. So here is the olive oil. It is red, and that to me suggests that there is some chlorophyll left behind, after the extraction protocol takes place. And that, that's what gives it its characteristic color. I also have soy oil. My soybean oil was kind of more clear. And we can see that there's a little fluorescence there, but not much. And that would suggest that, there's little residual chlorophyll left behind. And the last one that I had in my upstairs was corn oil. And look how bright yellow that is. And so I always wonder whether, that is from the xanthophylls or something that gives the corn its yellow color. That pigment is then able to excite and then emit also just that bright fluorescence. And so something that would be cool would be to then take, I don't have any corn at home, but like our next step, so thinking about that scientific method, here's our data. Now we can use this data to start thinking about our next set of experiments. So I have corn oil here, I have soy oil here, I have olives here. What would happen if I took those fruits and vegetables and macerated them like I did this, and then looked at them using the True Blue tube to see whether there was fluorescence. And so that is, again, how the scientific process, this framework works, right? So we had a hypothesis that chlorophyll, when it was on the True Blue tube, would be able to fluoresce. We looked at several different plant samples, and then we went from there. And so I think that the next step would then be to see if these oils then corresponded with their plants, like if we were able to mash it all together. And so we have about 5 minutes left. And so I'm going to do my last quick demo, which is household chemicals. And so household chemicals often have fluorescent dyes added. And this is for a couple of reasons. It's to make the product brighter and more appealing. Oftentimes there'll be optical brighteners present. These are common in detergents like clothing detergent because they make your clothing look brighter and cleaner. And so like if this is a simple one. So, what you would have your students do is do some research on optical brighteners. We also see optical brighteners sometimes in beauty products. And so, if we have those beauty products, is the optical brightener present? Can we see that fluorescing? And, a lot of times these are, these molecules are safe. So these molecules are not, hazardous, but they do change the way our eyes can perceive, these different dyes. And so I just went around my house and grabbed some cleaners. You would want your students to do some research. So perhaps everyone would bring a cleaner in from home or from around the classroom. I'm going to start with household cleaners. So, you would want to research. This is, dish detergent. But the thing I thought interesting about this was if you can see specifically right here, it's really small, but it does say, you can't really see that great, but it says brilliant shine. So the thing I was curious about, so I'm going to call this C for Cascade. That marker is not dark enough. Where is my dark Sharpie? Here is one. I thought I had another one around, a big one. Huh. Who knows? All right, so let's do the C for Cascade. And so I'm just going to start dabbing stuff on my plate in this last couple minutes. So there's the Cascade. Here's just some, dishwasher detergent, just like regular dish soap. Does regular dish soap have chemicals in it that optically brighten? I have Tide Cold Water Clean. This was a little sample I got on vacation one time. It definitely says whitens and brightens. So does Tide itself have optical brighteners in it? Are we going to be able to see that with fluorescence? Why isn't my marker working? All right, let's try the blue. Dark. Did I already do the blue? Yeah, that is not dark. All right, let's do this. Tide. All right, and I'm going to take a pipette. I'm going to dab some Tide on here. All right, throw out that pipette. I have another detergent around. This is free and clear detergent, just, generic brand, but I use the free and clear for my kids because they have very delicate skin. I'm going to put that there. It's supposedly free of any additional dyes, but does that mean it's free of optical brighteners? Who knows? And then the last thing I'm going to do is I have a Tide to Go. So I'll call that TG. That's one of these sticks, these laundry chemicals where, you can use it if you get a stain. And so I'm going to put a little bit of that on here. And then let's try it. Let's see what happens when we put this on the True Blue. So which one of these do you think are going to contain optical brighteners? Let's put this here. All right, close that down, turn my light off, and turn it on. Okay, and so what we can see is, this is interesting to me. So the C, the Cascade, that actually, even though it says it brightens, it did not have any optical brightener in it. But here we can see that the just plain old generic dish soap does. So that's cool. So is it an optical brightener? Is it have something to do with the color, the dye inside? I'm not sure. Here, very clearly, let me move this over a little. Our Tide with brighteners, you can see that is glowing. Again, that is likely because of the addition of optical brighteners that brightens and whitens your laundry. Again, it brightens and whitens because it is fluorescent, so your eye picks that up and it picks up that reflected light, which makes it look brighter. And then here we have our free and clear detergent, and that free and clear has no optical brighteners in it, which I thought was interesting. Okay, so that was a quick analysis there. I'm going to try a couple more things and then we'll wrap up really quickly. So just hang with me. I wanted to try some beauty products. I have this Aveeno Radiant Moisturizer. I thought because it was radiant, maybe that means it has optical brighteners in it. I can see that mark. Yeah, you can kind of see that mark. So I'm going to put some of the Aveeno here. I have another moisturizer, which is got sunscreens in it, but it does not say brightening. Are we going to see a difference here? So let's try this. This is just Neutrogena. Oh, there, now my marker's working better. I have plain old sunblock. This is from my kids. Again, this is one, I picked it specifically because it says fewer ingredients. So I was thinking maybe this would not have anything in it. That might like be an additional like optical brightener kind of thing. So let me do just regular sunscreen here. What else do I have? I have this glowy sunscreen oil SPF stuff. The one thing you might notice is it's shimmery. So does it have stuff in here that will make it brighter? And then do I have anything else? Oh, you know, I'm going to add something. The last thing I'm going to add is completely unrelated, but I do want to add it in the interest of time. If you've ever had problems with plumbing, you may have used special dyes to be able to trace where the water is flowing, either out of your pool or your toilet. You know, you want to figure out where it is. So you would add this to your toilet and use a light to determine where it's coming out. And it's very fluorescent. You only need a very small amount of it to get bright fluorescence. And so I'm going to turn off my light again. I'm going to slide this last example in here and I'm going to turn it on. And again, I'm expecting that some of these brightening, I kind of expect the brightening Aveeno and that oil that's like the glowy oil. I kind of expect that those might have optical brighteners in them or something because they seem shiny already or they mention brightening. I definitely do not expect anything to happen in the kids' sunblock. And the water tracing stuff should glow bright green. I don't know what's going to happen with the Neutrogena. So let's turn it on. Okay, so that bright green is definitely drowning out everything else. So what you can see is the chemical that we expect it to glow is glowing to the detriment of everything else. So let me remove that. Okay, so we can still see traces of it behind. But what we can see is that our Aveeno brightening lotion must have something in it that is causing an optical brightening because it appears to be glowing. And I see some like light glowing in my sunscreen samples. We know that sunscreen is going to absorb UV light, so that perhaps is a result from there. So this is a pretty cool, this would be a place where your students could go, they could read the components and really analyze what is in some products that are at home and determine which ones may be optical brighteners and which ones aren't, and then do the test. So it's kind of fun to be able to explore those things. I really thought that body oil would glow, but it clearly does not. And that's how science works, right? We make a hypothesis. Sometimes it's right, sometimes it's wrong, but all of the data is going to give us some really good information. So I'm just going to end with my markers again because they're pretty and I love to show the fluorescence. I'm going to do a quick summary. If you haven't, there I had a animation there that we forgot about. But oh my goodness, it's not even advancing through the slides. Let me go back to here for my wrap up. And so thanks again for joining me. If you have any questions, put them in the chat now while I'm wrapping up. But the scientific method describes a logical process that researchers use to answer questions in the lab. And while, we might, and here, let me put this GFP plate here too, just because glowy is cool. And so while, researchers may not follow the exact scientific method every time they're doing an experiment, I think it's important to realize that it is a framework that allows us to look at the research that's been done, to build upon it, and to ask questions, and then to move forward and to continue to contribute to the body of scientific knowledge. It is the progression through the steps, which then informs our next round of experiments. And simple experiments, like we did today, where we're just kind of throwing a bunch of stuff on our True Blue 2, which is our blue and white light transilluminator. These experiments are simple and they are fast. But the thing is, they can allow us to do these demonstrations that can help your students start thinking about experiments and learning about the process through which we research, we evaluate, and we analyze our data. If you want to learn more about the True Blue 2, it's on our website. I'll definitely link to it in the email. So if you have not filled out that form, be sure to fill out the form, and then I will be able to send you the slides, and I'll send you the information. Thank you guys for being here with me through this kind of grab bag of things to try. For me, it's just kind of fun that way, to really explore and, kind of just like today was kind of a grab bag, like I said, like just us exploring together and trying a bunch of stuff. We will be posting the slides here on YouTube. They'll also be on our website at edvotech.com. If you want us to email you when they're available, please fill out the form and we'll contact you in a few days. And I just want to say thank you so much for your time, for joining me. Sometimes things go smoothly, sometimes not. I think this went pretty well for something that was completely experimental. So I'm glad you think your students will love it. I'd love that they can ask real questions this way. Thanks again. While I'm here, I can pull up the link real quick for the True Blue 2 and put it into the chat so that you can take a look. But it's always fun for me to be here with you guys and with you people, all the people who are tuning in either live or in replay. If there's any way we can help you, please reach out to us by phone, by social media, by email. We want to hear from you. If you do try this and you get good results, let us know on our social. We'd love to hear it. Thank you so much again. Have a fantastic day and happy experimenting. Bye now.